telling us about integrative and parallel approaches to uncover drivers of cancer.
Thank you. I'd like to thank the organizing committee for giving me this opportunity to talk to you guys. And I want to talk about what I did at my last project in my postdoc. I've started, which was at Columbia University, and I've started at McGill. So one thing I want to start with a definition. When I say driver genes, I mean genes contributing to the growth or phenotype of cancer, either by presence, usually oncogenes, or by absence, usually tumor suppressors. I'm not going to distinguish between initiators, maintainers. I know that it feels like a lot of people have a lot of definitions, and it's good to focus on what I mean when I say that, just when I start. So basically, brief review, I use DNA and RNA together, where we have genome-wide measurements available, and integrating them actually gives us better results than looking at either. I'm not using protein data since, unfortunately, genome-wide measurements aren't available, but we've seen wonderful work how people can do it even if it's not genome-wide. So basically, when you look at DNA and you're looking at copy number, every gene you see tons of aberrations. You see, if we're talking about mutations, yes, but you're also talking about total changes in copy number, amplifications, deletions, and every patient has different combinations. If we look at actual data from brain tumor patients, TCGA glioblastoma, each row is a gene basically sorted by location. Each column is a patient. We can see that there are regions that are amplified in sort of all patients, and there are regions which are deleted in all patients, but almost at everything else you can find like a region that's deleted in one patient, amplified in another, or you can find regions that are deleted just in one patient or amplified just in one patient. And each patient seems to have a completely different sort of fingerprint. So first step when dealing with something like this is that we want to identify which aberrations are significant. And the biological principle that we're going on is cancer is aberrations in the genome are a random process under selective pressure. Therefore, driver aberrations that contribute to cancer growth and phenotype should appear in lots of samples in the population. And we use a modification of the logistic algorithm originally published by Baruchim et al. 2007, where basically many samples is statistically significant using an empirical null p-value distribution. Now, once you've gotten these regions, and they're unfortunately relatively large, and which gene in the region is their driver? So to that, we go back sort of to the central dogma that DNA causes RNA. And basically, if we have a change in copy number, that will change the expression. This is an actual example of an oncogene in melanoma called MITF, where you can see that, in general, amplified samples are overexpressed, but there are overexpressed samples that aren't amplified, and this leads to the phenotype. If this is a true hypothesis, then there might be other ways that cancer could change the expression, such as, and we've heard some of them, mutations in promoter regions, changes in upstream, changes in 3' UTR, 5' UTR that aren't necessarily related, and so on. So basically, if the gene is a driver, cancer will find other ways to over or underexpress it, so we probably need to look at expression. Now, going back to MITF, this is actual data where these are known MITF targets, which were isolated by ChIP-seq, and these are the exact same targets. The only difference is sorting the samples based once by MITF expression and once by MITF amplification. We can see that, yes, when MITF is amplified, these targets are overexpressed, but it's not very consistent when it's not amplified. If we sort by expression, they're extremely correlated. Now, you might be saying, wait a second, what you see from expression, this gene is correlated with this gene. How do you know which one is the driver? Well, that brings us back to DNA. My hypothesis is that since MITF is amplified and these genes are generally not, then MITF is a driver, these genes are targets. So that's actually great when I know the targets, but again, the problem is, how do I find the targets? So what I do is I use mutual information. I take the expression of driver, and I look for all genes, I'm showing 3 for simplicity's sake, if they have high or low mutual information. All the genes which have high mutual information are tentative target genes. And then I try to cluster them to find expression patterns that are consistent, because these seem to indicate a more biological function. So we can see these are all targets of a specific driver, and we can see that there are some specific expression patterns. That results in multiple drivers where each has a group of target genes, and the groups can overlap. We can see that genes 1 and 2 appear in both, are targeted by both drivers. And that can teach us about the function, standard gene ontology, and also the overlap between targets can group the drivers into clusters. So if driver 1 and driver 2 in this diagram, their targets overlap, we think, okay, driver 1 and driver 2 have a similar function. And actually, all 4 of these drivers are one cluster, where driver 5 is a different cluster. Now, if we look at sort of opposing overlap, i.e., genes induced by driver 2 are repressed by driver 6 and vice versa, well, you can turn on a relationship between the clusters, and you can say, okay, cluster 1 and cluster 2 seem to have an opposite effect on each other, or opposite effect in the biological network. When I did it with actual glioblastoma data, I ended up with about 80 driver genes that grouped into 4 clusters. I'm only showing a small amount of names, but basically it seemed to be proliferative, genes that are contributing to glioblastoma proliferation, genes that represent activation or aberrations in the PI3K pathway, and genes that represent the mesenchymal subtype and the proneural subtype, including known genes representing each type that are known to drive the relevant types, and mesenchymal and proneural genes proneural clusters were anti-correlated or seemed to be co-repressing, where the proneural subtype lowers the mesenchymal subtype and vice versa. This also matches the literature. Now, so far I didn't just discover existing drivers. We also focused on one gene, which is RHPN2, a novel driver gene. And RHPN2 is, here are the target genes. They're enriched very much for the mesenchymal signature, which represents invasion and generally has the worst clinical phenotype. We can also see this represented when we look at clinical data from the patients, where we have high RHPN2 leads to low survival, and low RHPN2 leads to high survival. Then we went to our biological collaborators, and we asked them to overexpress RHPN2 in a cell line that expresses it at a low level. Proliferation didn't change, but what changed is invasion and colony formation. They both went up in a significant manner. Similarly, when we knocked down RHPN2, we saw a decrease in invasion and colony formation, no change in other phenotypes assays. And interestingly, I hope you can see, if you overexpress, these are the controls, if you overexpress RHPN2 in cells that can differentiate into neurons, then they express more mesenchymal phenotypes, and when you try to induce differentiation, you get less differentiation. These are the figures, these are the graphs. So it affects them both phenotypically in multiple ways, and it affects their differentiation. So it seems to be exactly the biological effect predicted by multireg, and we were actually quite happy that our collaborators tested proliferation and got a negative result. At first, our collaborators were like, okay, fine, well, we can't put it in the paper because it's a negative result. We were like, no, this is a great negative result, because a lot of people check proliferation as their only screen. And we said, look, based on the targets, this is going to relate to be adhesion, not proliferation. The fact that it's not related to proliferation is actually a great negative result, if you want to phrase it that way. Because it means that if somebody had said, okay, I'm going to do an RNA screen of glioblastoma and look at proliferation, he wouldn't have picked up this gene at all. And our collaborators are pursuing it further and seeing if there are some drugs that seem to inhibit its mechanism of action, and perhaps can it have effect in mouse. So if we look at the targets and we combine multiple data types, we can look beyond proliferation, and this is an illustration from the hallmarks of cancer, which shows that, again, cancer has more than just proliferation. So, summarize, hopefully I'm not over nor under time. Great. I'd like to thank my postdoc mentor, Dana Paer, people from the PR lab that helped me with this project and other projects, especially a big thanks to Felix Sanchez Garcia and Helen Kalston. I'd also like to thank my collaborators, Antonia Verona and Carla Danussi, where Carla spent hours in the wet lab doing all these validations. I'd also like to thank Levi Garay's lab, I don't know if he's still here, that actually did the original data on melanoma, which helped us develop this whole thing. I'd like to thank the Charles H. Revson Foundation for funding my research. And before I take questions, I'd like to point out that I'm just starting my research group at McGill University, so I'm looking for programmers, students, postdocs. Please email me if you're interested. And I would like to thank you all very much for listening. Any questions?
So you talked about how you were trying to find driver mutations of cancer, and you also talked about how proliferation isn't the only phenotype of cancer. This gene that seemed to have a driver mutation was actually something related to invasion and not proliferation. So I guess another question you can then ask is, what is the driver phenotype of cancer? What phenotype shows up first? So do you think it's proliferation or invasion or something else?
I think I need a lot more time series data than data from TCGA, which is more snapshots. But based on what I've read, what I've seen other people published, my guess is that, and this is a guess, is that the first step in cancer is sort of some version of immortality, which is then followed by proliferation, which is followed by everything else. But I could be wrong about that. At this stage, it's an opinion. I think it's very nice. I have seen people who are doing sort of similar to what I'm doing, gene expression signatures, and they're trying to look at multiple types of cancers and seeing if they converge. Is it if you start with a cancer who's, let's say, if one cancer, in one cancer type, more glycolytic cancers are more, have a worse prognosis. In another cancer type, more proliferative cancers have a worse prognosis. Are they converging to some kind of very bad cancer in general, or is it just a thing where one type is dependent on this phenomenon, one type is dependent on this phenomenon, and they're not at all related? I'm not sure. I have colleagues that are doing this, but that's my guess. I don't actually know. It's an interesting question, but I don't know.
All right,
Thank you very much.
Thank you.